Have you ever wondered how plants communicate? It's not by their leaves, flowers, branches, or trunk, it's from their roots. Plants might look like they're not doing a whole lot of anything at any given moment in time, but in fact they're communicating in dynamic and complex ways with their below-ground ecosystem. In fact, plants can release up to 20% of their photosynthetically fixed carbon as root exudates. These root exudates are essentially the language they use to communicate with fungi, bacteria, archaea, and protists that live on, in, or near the roots. These microbes and the roots are collectively called the rhizosphere. The interaction between microbes and plant roots is a rapidly growing field of research, having largely been overlooked in the past. Two of the key findings from this growing body of research is first, the microbial communities are impacted by root exudates from the plant, and the second is that microbes can help improve plant tolerance to harsh environmental conditions such as salinity, drought, flooding potentially, nutrient limitations, and other stressful abiotic factors. It can help fend off pathogens, and it can increase crop productivity overall. So obviously all of these microbial benefits that I just listed are super applicable to agriculture, which, as we know, is both highly affected by climate change and is a key contributor to climate change. And this raises an important question. If plant roots can influence microbial communities through root exudation, can we harness this process to improve crop performance in the context of climate change posing a significant risk to the safety of our food systems? Now, to go back to the basics for a minute, what are plant root exudates? Root exudates are a complex mixture of compounds released from the plant roots into the surrounding soil. There are four general categories, the first being low molecular weight compounds like sugars, amino acids, and organic acids, the second being high molecular weight compounds like proteins and mucilage, the third being secondary metabolites, and the fourth signaling molecules such as flavonoids, which we'll talk about later. These exudates serve two major roles, the first being as a food source, so supplying carbon and nitrogen to soil microbes, and the second is acting as chemical signals influencing the behavior and interactions of microbes and plants. An important thing to keep in mind is that plant root exudation is not a passive process, as in it is not occurring at all times. It is a tightly regulated process. So for example, younger plants release more sugars and organic acids to support microbial colonization of hopefully beneficial microbes, whereas as plants mature, they will start releasing more secondary metabolites which recruit defenses against plant pathogens. Exudation is also highly variable and is influenced by plant species and genotypes, where we see some similarity in exudate profiles between general plant groups, but a lot of interspecies variation depending on genotype and a whole bunch of other factors. Soil condition plays a role in the exudate profiles, as well as environmental stress such as nutrient limitation and drought. The root type, even on the same plant, different types of roots will have different exudation profiles, and even the position along the root, with highest exudation happening at the tips or the base of the root and less so along the root. This means that the quantity and composition of exudates is highly variable in time, in space, and based on abiotic and biotic factors. Microbes matter in agriculture because they provide several key benefits to plants. For instance, some microbes such as rhizobium are capable of fixing atmospheric nitrogen, or N2, into nitrogenase, which they can supply to plants by forming a symbiotic relationship in the plant root and forming a nodule, which many of us have heard about before. Some lesser known examples are the mobilization of phosphorus by bacteria like Pseudomonas, which release an organic acid that can dissolve insoluble phosphorus that is in organic matter or in another form in the soil, which makes it available for plant uptake. Mycorrhizal fungi as well will extend the root system by essentially acting as hair extensions of the root system, making it easier for plants to take up nutrients like phosphorus from beyond where the typical root zone would be. There's even examples of some bacteria that can produce, or microbes in general, that can produce plant phytohormones. So for instance, bacteria like Azospirillum and Pseudomonas can produce plant hormones such as indole-3-acetic acid, or IAA, which stimulates root growth and increases the plant's ability to absorb nutrients. Certain microbes can even suppress plant pathogens by producing antimicrobial compounds or by outcompeting those harmful organisms. Again, certain microbes such as Pseudomonas and Streptomyces produce antimicrobial compounds that can directly inhibit the growth of plant pathogens. Beyond that, some microbes, like Trichoderma, can activate the plant's immune system, priming it to respond more effectively to future pathogen attacks, and this is evidence of a phenomenon we call plant priming. Because of these benefits, there's been a strong interest in using the microbial community, or microbial inoculants, in agriculture. So these are products that introduce microbes into the soil or as seed coats before planting. However, there are certain issues with these approaches because they often give inconsistent results. So introduced microbes can frequently fail to establish because they have to compete with native microbial populations and communities that are already there, and they can also fail to adapt to local soil conditions. In addition, there are some concerns about releasing potentially foreign microbes into a soil and what that'll do and how far they'll spread and how that might impact other plants. So this has led researchers to consider an alternative approach. Instead of adding microbes directly into the soil, can we enhance the plant's natural ability to recruit beneficial microbes? So, as we've established, some microbes are awesome and plants can potentially recruit them via root exudation, but how exactly does that work? So, as we established, root exudates play a central role in shaping the composition and function of the microbial community. One way they do this is by acting as a selective nutrient source, but this can be highly dependent on soil type, location, and a variety of biotic factors. So if we're trying to engineer the plant, that might not be the best, or engineer plant recruitment of microbes, that might not be the best approach. But, as we talked about, exudates do a lot more than just provide food for microbes. They can also function as signals and even go as far as regulating microbial gene expression, which I think is crazy. It's like a very strange form of mind control. So recent research has actually shown that plants can actively influence not just what microbes are present, but what those microbes end up doing over time. Let's look at a little case study. In the study titled Listening to Plants, Esperanto via Root Exudates by Feng et al., researchers work with two well-characterized plant growth-promoting rhizobacteria, the first being Bacillus belizensis, SQR9, and Pseudomonas protegens, CHA0, and examined how their gene expression changed across plant development. So to do this, they collected exudates from the plants at one species of plant, at different developmental stages and analyzed their chemical composition. From that, they identified 16 specific exudate compounds that varied over time. They experimentally exposed these bacteria to the stage-specific exudate mixture and individual compounds and used high-throughput qPCR to measure changes in the bacterial gene expression as a result of that exposure to those compounds. What they found was that changes in exudate composition directly drove shifts in microbial function. Early stage exudates from young plants activated genes associated with plant growth promotion, while later stage exudates from older, more developed plants induced genes related to biocontrol and stress resistance. This means that the plant wasn't just recruiting new microbes, it was chemically reprogramming the same bacteria over time by altering the signals it released through the roots, which is crazy. Importantly, another study expands on this by directly manipulating the composition of root exudates. So as I mentioned, one of the influences on root exudate profile is the genotype of the plant. So they used different sorghum genotypes. Researchers from that identified natural variation in the exudate chemistry and then recreated the profiles as synthetic exudate treatments. Specifically, one of the genotypes produced a high sugar treatment and one of the other genotypes produced a high organic acid treatment. So they applied these treatments to soil microcosms, which are essentially plant-free soil systems, so that they could isolate the effect of the exudates alone. By tracking microbial communities using 16S sequencing and metabolomics, they showed that different exudate compositions drove distinct microbial community structures and metabolic outputs. So for example, the high sugar exudates led to the enrichments of microbes associated with amino acid production and even phytohormone synthesis, so those plant hormones we were talking about, which demonstrates that exudates can shape not just who is there, but what metabolic functions are performed in the soil. Together, these studies show that plant genotype and developmental age can impact the gene expression, which could be the function, interpreted as the function, and the composition of soil microbes, which is huge. So based on what I just talked about, which essentially showed that root exudates are able to influence microbial community and microbial functioning by manipulating their genes, it makes sense that several strategies have been developed to manipulate root exudation. The first group of techniques we're going to look at is genetic and molecular approaches. One approach is to directly modify the plant's ability to release those root exudates. An example of this is to alter the transporter proteins, such as those in the ALMT and MATE transporter families, which control the export of metabolites from the root cells. And now it's case study time. In a study conducted by Kawasaki and al., researchers specifically manipulated the expression of these transporters in rice and wheat using transgenic lines and naturally varying genotypes. By doing this, they were able to control the release of exudates from the root apices, which is essentially just the root tip, or the part of the root that's still growing. So what occurred is when they overexpressed ALMT transporters in rice, this led to an increased release of malate, while variation in the MATE transporters in wheat resulted in increased citrate exudation. So using these modified plants, the researchers planted them in soil and used sequencing approaches to track how microbial communities responded. They found that by changing the transporter activity and therefore the composition of the exudates, they significantly altered both bacterial and fungal communities along the root systems, including at both the root apices and base. And remember, there's no exudates coming out of the base, so this is really interesting. But these effects were not uniform. The study showed that different root types and positions along the roots and soil conditions had a stronger influence on microbiome structure than the exudates alone. So this highlights a really important limitation we need to keep in mind when using these techniques. While transporter manipulation provides a powerful way to steer the root microbiome, its effects are context dependent and spatially variable across the root system. Overall, this work demonstrates that by targeting specific transport proteins, we can genetically tune the chemical signals released by the plants and thereby reshape the microbiome, at least to a certain extent. Although achieving consistent outcomes will require accounting for environmental and developmental factors. The second way that we could potentially manipulate exudates is looking at environmental manipulation. So this is a more practical approach where we can manipulate environmental conditions to influence exudation. So plants naturally will adjust their exudate profiles in response to environmental cues. For example, nutrient deficiencies can trigger the release of compounds that help mobilize nutrients. Soil amendments, such as organic matter inputs from manure, for instance, can influence exudation patterns as well. And crop rotation and cover cropping can shape long-term exudate dynamics. These approaches are potentially more attractive to use in a more practical sense because they can be implemented at a field scale and wouldn't require using specific genetic manipulation or planting of strains. So you could potentially prime your plants if you're expecting bad weather, if you're in a more controlled greenhouse scenario by exposing them to harsh temperatures or soil salinity or something like that. But this requires a lot more knowledge before we can move on. And now the glasses are coming back on because we're looking at a case study of where a lot of these techniques have been used. So we're going to talk about flavonoids now. One of the most well-studied groups of root exudates are flavonoids. Flavonoids are secondary metabolites that play a crucial role in plant microbe communication. They're especially important in initiating symbiosis with nitrogen-fixing rhizobia, as they are one of the first signaling compounds that goes out from the plant to the microbes, and in recruiting microbes that help defend against pathogens. Because of their various beneficial roles, there is an interest in increasing the production of flavonoids by plants. We've utilized some of the techniques I discussed above in increasing flavonoid production. These include abiotic stress induction, so conditions such as drought, UV radiation, which would mimic high sun conditions, or low temperature can increase flavonoid production. Nutrient deficiency, so nitrogen and phosphorus limitations, can stimulate flavonoid exudation to improve nutrient acquisition and microbial recruitment. Biotic elicitors, which essentially involves applying a signaling molecule like salicylic acid, which is a plant hormone, or introducing beneficial microbes, which can mimic stress signals and trigger flavonoid release. Soil management practices as well play a role, so organic amendments such as manure, which I talked about a bit before, have been shown to increase flavonoid exudation as well. And potentially genetic engineering approaches, which the two ways we found to enhance flavonoid production by genetic engineering is overexpressing transcription factors like PAP1 or modifying enzymes such as chalcone synthase or chalcone isomerase. There is a risk here though, because flavonoids are metabolically demanding for the plant to produce. So we've got to take things with a grain of salt. The most important takeaway from this section is that overall, these approaches show that it is possible to manipulate root exudates and therefore to influence the microbial community. However, achieving precise and predictable outcomes remains a major challenge in the field. Which leads us into our our next section, where we're talking about the limitations and challenges of this field of research. Personally, I think there's a lot of potential in this approach of manipulating root exudates for microbial or beneficial microbial recruitment. Otherwise, I wouldn't have done a project on it. But there are several important limitations to keep in mind. And this will also lead us into the research gaps, which we'll talk about in a little bit. The first is that root exudates are not necessarily the dominant factor in determining microbial community composition and functioning. Other variables such as soil type, root architecture, and position along the root can have an even greater influence on microbial community structure, which means that how we apply this technique will be highly site-specific. The second is that the effects of exudates are highly localized in the soil. They're strongest at the root tips because they're providing all that carbon to the microbes, but might not extend as far into the surrounding soil where the microbial community composition might be determined by other pools of organic matter. The third is that exudation is highly dynamic. As we talked about, it changes over time, across developmental stages, and in response to environmental conditions. And we still lack a clear understanding of when specific compounds are released and how this timing affects microbial recruitment, which is why I've been vague about some of the details in this video, because we just don't know yet. Another major limitation is that plants are constrained by the native microbial community present in the soil. So plants, in theory, can only recruit the microbes that are already there. If beneficial microbes are absent or present in very low abundance, then modifying root exudates alone may not be enough to establish a desirable outcome. You can't recruit what isn't there. This suggests that combining strategies may be more effective. So if you remember in the beginning, I was talking about microbial inoculants and why they're not always effective. Some research now is looking at using microbial inoculants in conjunction with this modified root exudation, which might help recruit and maintain those communities so they don't have such a short shelf life, if you will, in the soil. Finally, the most major challenge is that we don't know what defines an optimal or beneficial plant microbiome first. I've talked about some of the groups that we know are beneficial, like rhizobia, which only form symbiosis with legumes, and not all rhizobia are beneficial. PGPR, so plant growth promoting rhizobacteria as well, we know are helpful, but again, that's a limited part of the microbial community, and arbuscular mycorrhizal fungi are also useful, but probably not all species. So again, there's a lot unknown there. Without that knowledge, it is really hard to design targeted strategies with predictable outcomes in terms of microbial manipulation via root exudates. So what does this mean going forward? Future research needs to address several key gaps in this field. We need to better understand, one, which microbial communities provide the greatest benefits and how that changes under different conditions. Two, which specific exudates will recruit those microbes, which again is probably going to change under different environmental conditions and different soil types. And three, how exudation patterns change over time and across environments. There is strong potential in integrating multiple approaches like I was just talking about. So that could be combined plant breeding, microbial inoculants, and environmental management. Another important direction is identifying which transporters and pathways control exudation, which will be really useful to do once we figure out what root exudates recruit what beneficial microbes and how we can target that. Ultimately, the goal is to develop a framework that managers could use to improve their crop resilience with these techniques. But given that we have a hard time sometimes getting farmers on board with more simple and intuitive practices, and there's many complexities with microbiology as it stands, this might be a long road before we get to where we want to be. So in conclusion, this might be a little repetitive, but I've thrown a lot of information at you in the last few minutes. Through root exudates, we know that plants can actively shape the microbial communities around them. So this means recruiting beneficial microbes, defending against pathogens, and responding to environmental stresses. This represents a powerful opportunity for developing more sustainable agricultural systems, potentially from moving away from practices that we know are harmful, like the use of agrochemicals such as pesticides, synthetic fertilizers, herbicides, and all that kind of stuff. It might also be really important, as I talked about, in the wake of climate change, where we expect drought and soil aridity to be a significant issue. But this is a living system, so it's complex, it's dynamic, and is still not fully understood. There are two big gaps in answering the question of if we can manipulate root exudates to improve crop performance by recruiting beneficial microbes. And I will leave you with these two gaps and questions we need to understand. One, we need to determine the key components of the beneficial microbial community and what substrates recruit these microbes. Then we can potentially target transporters in the roots and increase the release of these exudates. Two, we don't know how exudate changes in cultivars, species, growth stages, environmental conditions, and therefore cannot clearly explain how most microbes are attracted to roots. We've talked about certain, such as rhizobia, but that's kind of a one-off and a clear example of a biosynthetic pathway and of a pathway of two organisms interacting that we understand, whereas most microbes we don't understand yet. So this also impedes further research. Thank you for watching this video today. I had a lot of fun working on this project and feel like I learned so much, and I think it's going to be really interesting to see how we develop solutions and systems to study this stuff. I mean, soil's been seen as a black box for so long, but I think a lot of attitudes are changing around that, and I think in the next few decades, hopefully in this decade, the next few years, we'll see some advancements in this research. Thank you.